the PhD dissertation defense of the English language. And yes, and second, our chairman of our dissertation council, Professor Bisimbayev Amagin is affiliated with Alžanұlы, and therefore I will be moderating this meeting today. So the first, we have to, as usual, introduce the invited members of the dissertation committee, and we have to at least introduce permanent members of the dissertation council, and therefore I will start with invited members of the dissertation council. First is Ahmetovayev Ilyas Amirkhanovich, candidate of biological sciences. He is from National Biotechnology Center, Astana. Ilyas Amirkhanovich, hello, you here? Okay.
Микрофон отключен у вас.
Сюда, здесь. Все, косынка.
Second one is Janusova Gulnur Sabundikovna. She is a PhD associate professor from the Institute of Genetics and Physiology. Gulnur Sabundikovna, добрый день.
Добрый день.
The next one is Karizhanov Roman Bogdanovich. He is a PhD associate researcher at the University College London in United Kingdom. Is that correct?
Master Maya, clinical research fellow, Institute of Neurology.
Okay. Thank you very much for joining today. And the permanent members of the council, we have a chairman here, Professor Bisimbayev Amagin Kwanbayevich. He is a professor at this university. I'm his deputy. My name is Toruspekov Yerlan Kenizbekovich. And third member of our community is Professor Plekhanov Sultan Plekhanovich, professor at this university, and the scientific secretary Nurmuratova Miranku Kudryatova. Thank you again all for joining today's meeting. We're a little bit late because of some technical issues. And also for this defense, we have two official reviewers. First is Akanov Janay Aykanovich. He is president of the Kazakhstan Society of the Study of Diabetes, member of the Executive Committee of the Asian Association for the Study of Diabetes. He's from Almaty, Kazakhstan. Janay Aykanovich, hello, добрый день, здравствуйте. Okay. And the second one is Yerezhepov Daurian Adilovich. He's a PhD at the Nazarbayev University. Daurian, hello.
Hi. Hello.
Okay. So today's agenda is defense of the PhD dissertation by Bahjan Alžanұlы, entitled, and his thesis entitled Development of the Cell Therapy Approach for Diabetes by Engineering Tunable Insulin Production in Beta Cells. Do you have questions related to the issue, to this agenda? Can we start the meeting? No questions? Okay, then let's start. The first step in our today's meeting is introduction of his personal information about the PhD student, Bahjan Alžanұlы. Please, ask scientific secretary Nurmuratova Miranku Kudryatova to introduce today's student.
Dear colleagues,
Здравствуйте.
can I?
Вас слышно, спасибо.
Спасибо, что присоединились. Thank you.
I'm so sorry that's for lating. I'm so sorry. That's at least that I have some resume of the work of the Bahjan Alžanұlы, So actually speaking, at least, I'm very happy that we have in Kazakhstan, we have the very good technology.
Professor,
Janay Akana.
thank you for the, we already introduced you. Thank you. We already started the meeting, so we will.
I'm so sorry for late.
No problem. Okay.
Let me introduce you with a set of documents contained in the personal files of doctoral student Alžanұлы and the information about him. Alžanұlы was born in 1991. Nationality is Kazakhstan. Place of birth is Jambul region, Republic of Kazakhstan. Education is higher. He graduated from biotechnology program at Al-Farabi Kazakh National University in 2013. Then in 2017, he completed a master's degree in biotechnology at New York University. In 2017, Bakhchan enrolled to PhD program in biology at Al-Farabi Kazakh National University. Scientific internship within the framework of the doctoral program took place between August 1st to late December in 2018 at the Diabetes Research Group facility of the University of British Columbia in Vancouver, Canada. The following documents are provided in the personal file of Alžanұлы. Application to the rector of Al-Farabi Kazakh National University for the defense of the dissertation, a copy of the minutes of the Academic Council of the Faculty of Biology and Biotechnology, and on the topic of the dissertation and the approval of scientific advisors. Copies of diploma or higher and postgraduate education, a copy of the transcript of professional study of the doctoral program at the university, dissertation of hardcover and electronic version, annotation in the 3 languages, review of domestic scientific advisor Sharipov Kamaldin Orunbayevich and the foreign scientific advisors Panzhinsky Evgeny Alexandrovich. So minutes of the extended meeting of the Department of Molecular Biology and Genetic approved by the Vice Rector for research and innovation activities with a positive resolution. List of scientific works approved by the Scientific Secretary of the Faculty of Biology and Biotechnology and their copies. The conclusion of the absence of violation in the process of planning, evaluation, selection, conduct, and dissemination of research results, which include the protection, the right, safety, and the well-being of research objects of the ESIC Commission of the university where the doctoral student studied. Reviewers or official reviewers, Yerezhepov Daurian Adilovich and Akanov Janay Aykanovich, certificate of anti-plagiarism by the National Center of Scientific and Technical Information, certificate of the Library of Al-Farabi Kazakh National University of the dissertation and annotations, and personal files certified by HR department of the university. The main content of the thesis is reflected in 13 printed works, including 2 articles and 1 thesis in foreign journals, with an impact factor and indexed in the Web of Science and the Scopus database with high percentile. 3 articles in national scientific journals, 2 of them from the list of the Committee of Quality Assurance in Science and Higher Education, and 1 article and 6 abstracts in the materials of International Scientific Conference. Presented materials by the applicant Alžanұlы comply with the rules and the requirements of dissertation for the defense by the Committee for Quality Assurance of the Science and Higher Education of the Ministry of Science and Higher Education of the Republic of Kazakhstan. Therefore, the defense is allowed. If you have any questions.
Okay, thank you very much. Any questions?
Actually speaking, dear colleagues, I want to explain my very high respectful to this doctor's degree. I want to explain any technology, any high technology in the fields on the diabetes type 1 is just a future. Hello to the Dr. Bahjan. I want to explain to him that is just one step. Next step should be future.
Oh, thank you so much.
We also have two scientific advisors for this PhD student. First is Sharipov Kamaldinov, Doctor of Biological Sciences, Professor, Director General of the Alhozan Institute of Molecular Biology and Biochemistry, and Dr. Panjinsky Evgeny Alexandrovich. He's a researcher at the Diabetes Research Group, University of British Columbia, Vancouver, Canada. So any questions to our scientific secretary? No questions. Okay. Then we have next step of our today's agenda. Alžanұlы, you may present your work in the given time is 20 minutes, please. Okay.
Hey, dear dissertation council, dear reviewers, and dear the invited guests, thank you very much for your time and attention today. It's my honor to be here and to be able to share my research results with you. Let me quickly
Поделиться.
share. И слайд. Okay. So I was given the floor to present my research, which is related to diabetes. Particularly, my research topic is called developing a cell therapy approach for diabetes by engineering tunable insulin production in beta cells. By beta cells, I mean pancreatic beta cells. I have graduated from the Department of Molecular Biology and Genetics, as was already said, and I'm also a research scientist at Alžanұlы Institute of Molecular Biology and Biochemistry. So I think it's undoubtedly clear for everyone who is related to medicine and science that diabetes is now putting a big burden on national healthcare of any country. So if we look at the human body, all the focus is on pancreas, the organ which is the organ which holds a very important function, and we today talk on the endocrine function of pancreas, which is secreting insulin, which comes as a very important and very important hormone. The insulin itself is produced by beta cells of pancreas. I mean the endocrine part of the pancreas, which is called usually Langerhans islets. So in beta cells during the diabetes type 1, the cells are distracted by immune system, the own immune system of the body. So as a result, we have absolute deficiency of the hormone, which, as I said, very important hormone, and which the lack of which leads to lethal case. So diabetes, if you just look at the numbers, is the worst scenario is that the numbers are growing very fast. So if we take as the last 5 years from 14 to 19, the number of diabetes death cases in Kazakhstan is already quadrupled. And if you check out the overall the number of patients, it's 13,000, which is considering the overall population of 20 million of Kazakhstan. If you consider that number, this number is quite high. And if we also consider that diabetes type 1 is usually 10% of all diabetes cases, then we can imagine that the overall diabetes case is pretty much pandemic situation. So so many researchers around the world, they are working on to come up with a solution for this disease, because right now there is no cure. All the diabetic patients, they have to inject exogenous insulin just to keep their lives. Otherwise, they will die. So many research approaches are being developed in the world in the last year. So in this research, considering the recent and latest advancements of genome editing technologies, and particularly CRISPR-Cas9, the purpose of my research was to come up with a genetic approach which is based on CRISPR-Cas9 technology to modulate the transcription of endogenous insulin in first human embryonic stem cells, and then differentiate those genome-edited stem cells to to pancreatic beta cells, and to see that any feasibility with keeping the modulated insulin expression in the obtained beta cells. So for that, I have come up distinguished 6 research tasks. The one which will be to design a lentiviral vector containing insulin guide RNAs to direct the CRISPR complex, and then will be 2 plasmids for activating and inhibiting insulin transcription in the first in the stem cells, and then in the differentiated pancreatic beta cells. And then, of course, we need to validate the constructs. So the third part will be validation. Then after we have validated everything in ordinary human cells, then we move on to stem cells. And after we see that in stem cells the constructs are working, we can now move on to the 5th research task, which is differentiation of genome-edited, CRISPR-edited stem cells towards pancreatic beta cells. And then the last research task is to study the features and properties of the obtained cells. But most importantly, we are more interested in to see what will be the glucose responsive function of the obtained cells. Just a very brief background on the CRISPR-Cas9 technology, how this technology came up, and how from bacterial immune system we went to leveraging this technology in eukaryotic cells. These 2 ladies, Jennifer Doudna and Emmanuel Charpentier, they were awarded a Nobel Prize in 2020 for discovering the technology which were based on bacterial adaptive immunity. So these 2 ladies, just by changing a few changes in the CRISPR complex, they were able to use that as a tool to modify the insulin, endogenous gene expression in eukaryotic cells. So instead of using Cas9, native Cas9, which is nucleus, which can cut any DNA sequence, they came up with deactivated form of Cas9, which is by inducing point mutations to the active site of the nucleus, they made the nucleus being deactivated. So now the nucleus can no longer cut any DNA sequence, but which is interesting is that the nucleus can act as a tool to bring needed transcription factors to any gene of interest inside the cell. So in this research, I proposed 2 mechanisms. One, as I said, I have here the CRISPR complex, which is dCas9, linked with the guide RNA, which is very important, which is complementary to the target sequence. So if we just link the Cas9 with a transcription activator and then use that complex, quite simple complex, to activate the insulin transcription inside the cell, while the second mechanism is just instead of VP64, which is synthetic transcription activator, we could use CRAB, which is also synthetic transcription factor, but in this case it acts as an inhibitor of the transcription. So in these 2 cases, we want to see how this complex is gonna work in ordinary human cells and then in stem cells, and if that would be feasible to modulate the expression. So if we just take out all the fancy images and then just to briefly say the workflow, the overall work consists of two parts. As I already said, the validation part, which is done on ordinary cells. In this case, I used HEC293 cells, which are very popular in biomedical research. So we prepare the constructs, and when we first transfect the cells with the first part of the complex, which is plasmids, and then we transfect the cells, go and transduce them with lentiviral vector, which contains insulin guide RNA for driving the CRISPR complex. And then we go and check the insulin expression. If everything works well, then move on to the stem cell part. The following, the order is quite the same, but only after we see that the constructs are working in the stem cells, we go to differentiate them and then assess the glucose responsive function of the obtained final cells. So now we are moving on to the results part. So here I see, just for brief understanding how the plasmids look like. On your part, we see the lentiviral based plasmid vector, which contains two insulin guide RNAs for driving the expression of the insulin inside the cell. So each guide RNA is driven by a separate promoter. So this plasmid then was used to package a lentiviral based vector to transform them inside the cell. On your left, we see, of course, I used several plasmids, but here just for understanding how it looks like, the representative image of plasmid carrying DCAS9 with BP64, in this case for activation, but instead of BP64, if we use CRAP, then it's going to become as the inhibition complex. So, as I said, first part of the work was validation in CRISPR cell in ordinary human cells. So, after doing all these transfection and transduction experiments, we see that comparing to control cells, which were not edited, the study cells, they show a quite higher level of insulin transcription activation. And if we compare it to natural Min6, the Min6 cells are natural mouse cell lines, which naturally produce insulin. So as a positive sample, we can still, compare them. Of course, the levels are not comparable, but still, we see that our study cells, they gave us the quite high expression of insulin. And then, as I said, this was the first part of the experiment with validation. The second part was using another transcription inhibitor, which is CRAP protein, to try to lower the already activated level of insulin in these cells. So when we introduce another plasmid inside the cells, then we see that the activated level was dampened quite much, and it was almost quite close to the control cells. So in this validation part, we can see that with all the two synthetic transcription activators, they are working quite well. And also, just to confirm the results, the fluorescent image of the cells with successful activation of insulin transcription. So at this point, we can say that the constructs are working, and the ordinary human cells, they were able to switch their activation, insulin activation inside the cell. Then the quite interesting part moves on to the stem cells. So the same constructs, the same order, we transduced stem cells. In this case, I used H1 line of embryonic stem cells. This is quite a popular and well-known line, and mostly used for gene activation studies in using stem cells. So for that case, to confirm the modulation of insulin expression, I used two methods. One was the classic qPCR, where we can see that comparing to human islets, we know the qPCR product size, which is 150 base pairs. For that, our activation construct was able to give some detectable level of insulin. Of course, it's not that much like in the case of human cells, human islets, but still we can see that these two samples, they gave the activated expression of insulin in these stem cells. And then another part was that I wanted for myself to confirm that the cells are really making insulin. So I moved on with a method which is called staining. There is a quite popular dye, which is called pure staining, which can really show us what kind of cells are expressing insulin and what kind of cells are not expressing. So comparing to the negative control and positive control, the activated and dampened samples, they all really in correspondence to the level of activation of insulin, they really showed which cells are really making insulin. So at this point, we can say that now the stem cells, they also started making insulin. So that's another part of the story. The next part is what I already said, the differentiation of these stem cells. But before going to differentiate, I just wanted to remind you quickly that while the embryo develops, we know that 3 layers of germ layer formed: ectoderma, endoderma, and mesoderma. And we know that the pancreas is formed from endoderm. So during the gastrulation gastrulation step, there is a definite endoderm cells. When the gastrulation is over, all the cells of endoderm layer, they are called definitive endoderm cells. And this is very quite well understood in science already. So now there are lots of protocols which can give us pancreatic beta cells from stem cells. I mean, no matter embryonic or induced pluripotent, but in this case, I used embryonic. So following the protocol that was earlier published by a group of Rizania in Nature Biotechnology, so the 7-stage protocol was used. And in each stage of the protocol, of course, there are key biomarkers which will be monitored, which will be used as a guide if we are on the right path or not. So at all the stage, there are 1 or 2 or 3 important biomarkers. So every stage, we will not stop until we get the desired activation expression of these biomarkers. We add many cocktails, growth factors, I mean, lots of repetitions, and then at every stage we collect the cells, we extract the RNA, then we go with qPCR and see, are we there or are we not yet? So this is quite a laborious part of the work, but it was very interesting for me to see if the functionality of CRISPR complex is going to be lost at any point during this differentiation. So that was interesting to see. And of course, the most important part is, are we going to get there to the last stage, where the pancreatic beta cells, they need to have clear 3 biomarkers, which is one important is, of course, insulin. And then there are 2 other very biomarkers which distinguish beta cells from other parts of the pancreas. There might be sigma cells and alpha cells, etc. So that's just to wrap up the protocol. So now I'm moving on to the results of every stage. We can see that there is a representative image of the cells, how they look like just before the start of the differentiation. The confidence was around 85%. They were cultured for 48 hours in the E8 medium. So now then we move on to each step, and each stage of the cell, we know that what kind of biomarkers we are going to see and what kind of important growth factors we are going to be adding on each stage. Of course, there are only a few of them, but in reality, there might be 20, even 30. There are so many. And on each step of the differentiation, we will be measuring the expression of the key biomarkers. And the important thing is that all the stage, all the day of the differentiation, the biomarkers expression, they're going to be rising. I mean, if they steady or if they go down, then which means that the differentiation is not working well. So the idea, the goal is to maintain the growth of activation of every biomarker. So this is the stages from 1 to 3, quite simple, but now it gets more and more complex. So here we see the differentiation stage number 4. Then now the 2 biomarkers, this kind of, just for example, this kind of growth factors cocktails were added. And as I said, all the time we collect seeds, we extract RNA, we compare seeds, cells with each other, and then try to see that maintain that the growth of expression biomarkers is maintained in all the stages. And when we go to the differentiation stage 5, we have 3 biomarkers. There are also biomarkers the level of which needs to be going down, and some biomarkers are going to disappear. I mean, they're also important, but the main biomarkers are these ones. And then we go to the stage 6, which is quite close to the end. At this stage, cells start to express insulin, some insulin. You can see the insulin. The longevity of the stage is quite high, I mean, 12 days, 2 biomarkers. And at this stage, we also need to look for other hormones, which are usually made by other cell types. I mean, glucagon by alpha cells and somatostatin by other cell types. So their level is going down. I mean, at this point, we can see that we are on the right path, and we are very close to complete the differentiation, which is the last final stage, stage number 7. All the needed 3 key biomarkers are maintained. The growth is also maintained as well. And the final pancreatic beta cells with clear 3 biomarkers, distinguished biomarkers. And at this point, we said to ourselves that we are done with the differentiation. So that's another part of the story. The next part of the story is, okay, we got the pancreatic beta cells. So how the insulin expression is going to be there? I mean, we know that in the beginning of the experiment, everything was clear. Now we wanted to see what about the insulin expression. So comparing to human islets, all the time human islets, I mean, natural expression, we wanted to see our study cells, and for our fortune, we see that some level of insulin expression is still there. I mean, quite detectable, quite significant, still there. With the negative control, which lacked in guide RNA, there is no expression left. So that was very fortunate and very good to see. And then there was also, inside the study cells, I made a comparison between ordinary stem cells, which are not CRISPR edited, and another group is CRISPR edited stem cells. So in CRISPR edited stem cells, insulin expression was mildly higher than the non-CRISPR edited H1 derived beta cells. So that was the comparison. That was the pre-last part of the research. And the last part, was I showed in the beginning, is to assess the glucose responsiveness. I mean, this was not really the question of the dissertation, but that was interesting for me to see how they're going to act, because no matter what kind of very good cells we obtain, if they are not going to respond to the changes of glucose concentration in the media, then there is no good from the cells. I mean, because in nature, in our body, the pancreatic beta cells, they can see that the person had a food, now the glucose level went up, now they are going to respond. So that was mimicking the real case. So in the in vitro, I took my study cells, I compared them with human islets, and then also compared them with non-CRISPR edited, but still H1 derived beta cells. And I see that there are some detectable level of glucose responsiveness is there, but the reality is that comparing to natural human islets, the numbers are not, I mean, comparable. It's quite high. The difference is quite high. But what is good is that using CRISPR technology, we can modulate insulin expression in stem cells, then differentiate them towards pancreatic beta cells, and keep the effectiveness of the construct, which at the end showing us that is also affecting the glucose responsiveness function of the final cells. So that was the end of the story. I used the three levels of glucose concentration to see how they're going to act for that. Here are my conclusions. As I
familiarized with your conclusions, and I think we should now switch to the questions. Dear colleagues, specifically invited members of the dissertational council, we are switching to the questions. Please ask your questions. Please raise your hand and so we can see you and your willingness to ask questions, please. Anyone can ask questions, including official reviewers. Вопросы можно задавать и на русском, и на казахском, и на китайском языке, на каком хотите. Пожалуйста, коллеги, мы сначала хотим послушать именно специалистов, приглашенных специалистов, чтобы полнее раскрыть работу. Пожалуйста.
I have a questions. Can I ask?
Yeah, sure, you may start.
How did you design and validate your guide RNAs in your experiments?
Should I go ahead?
Yeah.
Okay.
In short, could you explain?
Yeah, sure. Sure. Yeah, I think that's a very good question because the guide RNAs, during the experiments, I made some changes to see that some CRISPR complexes without guide RNAs, they don't work. So for me, it was very important to validate the guide RNAs. So since the CRISPR technology, the field is evolving very fast, there are already some softwares which help us to design guide RNAs. For example, in my case, I use the software which is called Guide Maker or Maker Guide. So it's so if you just input the target sequence, and the software is going to design for you the several options of guide RNA. So that's the first part. The second part of the answer is I need to validate guide RNA. So for that, I used the human islet cells, and I constructed a real active nucleus Cas9 with guide RNA. And then when I inserted the complex inside the human islets, the guide RNAs, they really drove the nucleus to the target sequence, and at the end, the human islets, they stopped making insulin, which showing me that which guide RNAs are really working. So how they are going to be able to link with Cas9 and then drive the target sequence. So that was the way how I designed, and then we validated guide RNAs.
Sorry, Bakhjan, can we ask you to talk to the laptop? Because when you're talking to the auditory, we doesn't hear very well.
Okay.
So you have to talk looking at the laptop, because I think you're talking to a laptop microphone, right?
Yes, I'm using the laptop's microphone.
Yeah. So.
Gulnur Sarnandikana, do you have some more questions?
Yes, I'm thinking still. So thank you for the question or for the answer. It's very clear answers I got. So maybe I have second questions, but so what kind of limitations are there that might be possible stop applying your results to the clinical stage research?
Limitations.
Yes, limitations that might be possible stop applying your results into the clinical stage of the research.
Yeah, thank you. That's a very good but very conceptual question. So I can answer on two things. First of all, this is still in vitro stage. So even the in vitro stage is not completed yet, because we see that even though the glucose responsiveness, I mean, assessing that property of the cells was not part of the dissertation, but still in order to move forward with the development, the cells need to be clearly responding to the changes of glucose concentration in the media. I mean, I'm far from that yet. That's the one reason as a limitation. And second reason is that in this case, I'm using embryonic stem cells, which already, by it means might cause questions in terms of ethics, because embryonic stem cells, they can be used in researchers for no more than 2 weeks. So in this case, I'm using embryonic stem cells to have better final differentiated cells. But if I want to move on with the research, then I need to be using induced pluripotent stem cells. For that, there is no limitation in terms of ethics. So in my case, using embryonic stem cells is also additional limitation.
Thank you. Gulnur Sarnandikana, are you satisfied with the answers?
Yes, I satisfied. I can understand. Thank you for the answering.
Thank you.
Now I got
I wonder if Roland Bogdanovich has any question for Bahjan.
Yeah, thanks a lot, Bakhchan, for a very informative, short presentation. I think it was quite understandable. It's quite easy to follow, even for people with non-biology background. So I have a few questions. So the first question is just related to the previous questions, to the previous question that our colleague asked. So in terms of translation of your findings to clinical practice, what predicaments you would kind of anticipate, even if you worked on induced pluripotent cells, no iPSC cells, and what you can tell about like cellular environment, because what you have done is in a cellular models, but would this nicely translate into human cells, which will have a different environment?
Mm-hmm.
This is the first question. Thank you.
Yeah, okay. If I, yeah, I always think about the future. So if there are lots of, in fact, there are lots of limitations in my study, because the study first is more on like a feasibility study. Feasibility by means like, if it's going to be able, the construct is going to able to allow us to change, to modulate the expression of insulin in stem cells, and then what's going to happen after differentiation. So if I use the induced pluripotent stem cells, I can think that I can still be able to differentiate them towards pancreatic beta cells, but since induced pluripotent stem cells, they are less stable than embryonic stem cells. I mean, for that, I'm actually using embryonic stem cells. I mean, the final cells, they may not be able to have insulin expression at all. I mean, that's one thing. And if there is no expression, then of course we can't even talk about the glucose responsiveness. And in terms of differentiation, it's also a question that I might be, even if I'm going to be able to complete the differentiation, because for induced pluripotent stem cells, the protocol differs slightly, but they are less stable, and there is a higher chance that during the differentiation, the effectiveness of the construct, they might be lost. So lots of questions and lots of, if we can say, limitations maybe in front if we use these pluripotent stem cells.
Thank you, Bakhchan. So the question was also related to the cause of diabetes type 1, which we know which is complex, involving autoimmune and environmental and complex genetics, right? So do you think even if we use these iPSC cells, but how we can protect it from autoimmune response, in terms of immune compatibility, as well as kind of a possible tumorigenic risk of CRISPR and Cas9 editing? How far we are from implementing these cellular findings and cellular results into real human body, do you think? How far we are?
Yeah, thank you very much, Roland Bogdanovich, for this interesting question. I see that you are looking in terms of the application of this, I mean, overall advancements in this field. So from the, as someone who is monitoring the field, I can say that we are not quite far from seeing the leveraging this type of, no matter with which tools, I mean, the healthy, ready to be transplanted, the healthy pancreatic beta cells. I know that there is a company, world company, which is called Vertex. They are really advancing the field, and they know that they have two solutions for this. In one case, since you already mentioned the autoimmune ability, so they are using artificial shield as a protector from the attack of immune system. So the cells that they came up, the cells are shielded inside the artificial organ, which protects them from. Until now, they are studying this, we can say, device or whatever in a phase 2 clinical trials. So since the clinical trials, they have already started, this is my personal opinion, that we are not quite far from that. But of course, how far, we can't say exactly yet. In terms of tumor progression, of course, it always remains as a threat. So I know that the company, the Vertex company, they shared the latest updates in spring of this year. And there are some cases, some patients were withdrawn from the trial because they had some health problems. They don't say exactly what, might be possibly because of tumor, but they have already started feeling the adverse effects of this trial. So, of course, including tumor, there might be some questions that might come up during the past. So it's always a challenge.
Thank you, Bakhshan. I'm satisfied with this answer. And if it is okay, I have a couple of more questions related to the experimental part of your work. If it is okay, dear committee.
Please go.
Yeah. So the question about cellular mosaicism. So when you introduce this CRISPR-Cas9 guide RNA, did you consider any kind of do you think this mosaicism was related or was relevant to your experiments? And first question. The second question, I'm not expert in HEK cells, and because I'm mostly clinical researcher, but with a methylation profile of these HEK cells and the human embryonic cells should also be considered in your experiments, just to get an idea. And in slide 9, you showed several biomarkers, and I understand these are genes, right, that you targeted, right? These are genes that you targeted with the guide RNA. And so you mentioned several, like PDX-K1, NKX61, and what about other genes, like other important genes that are responsible for insulin production? Did you target all of them, or why did you specifically choose several biomarkers as a result of your literature review? So these are my questions. Thank you very much.
Okay, so if I start from the last question, in terms of differentiation, so there is no proper, very good, and accepted with everyone. There is no protocol yet. Lots of groups are working on it, and the protocols differ a lot. So I see that your question is coming from that point of view. In my experiment, I mostly stick to follow the protocol that was published in 2014 by the group of Britannia and others in Nature Biotechnology. So in their protocol, they also advanced it to the in vivo study, to my study, but in the in vitro one, they came up and developed this protocol. So they specifically targeted those biomarkers. So in my study, I also did this. Of course, it's not came out from the one repetition. There were a number of repetitions, but all the repetitions showed me that following this path can drive me to getting pancreatic beta cells. I know that in some protocols, the biomarkers they differ. The growth factors cocktails are also differ. So who knows? There is also quite interesting thing that the race is ongoing. Who will come up with a very good protocol and who is going to get Nobel Prize for that? So in terms of that, this is also still discussable, debatable area. But in my case, the protocol did a quite good job, and I was able to get the pancreatic beta cells. I forgot what was the first question you said. You mentioned
it was related to mosaicism, cellular mosaicism, and high methylation profile of the genes that you targeted in these cells. Do you think it's relevant to your experiments, and would it somehow affect the results of experiments if you say consider the baseline level of methylation of these genes that you initially targeted? Would it be relevant or not, just to get an idea of your thoughts on this?
I think that in my case, I didn't really pay attention to that because there was lots of factors. There was really time constraint. I was in Canada, and I was only quite a few months left, so I was trying to expedite the experiments and see the stem cells and do the experiments by my hand. So of course there are lots of aspects, issues that might come up. They might also have a big role in the final result. So in that case, what you mentioned is really important, but I haven't really, honestly, I haven't really had a chance to pay attention and to look for what kind of other issues, other profiles they might also affect. So who knows?
Thank you. I'm not bringing it up as a limitation of your studies. I just wanted to have your idea, or what you think about this. Yeah, but not. I don't. Please don't consider this as a limitation of your study. Okay.
Yeah, thank you.
Thank you very much. I'm satisfied with the answers.
Thank you very much. And I wonder if Ilyas Amirkhanovich may have some questions.
Добрый день, коллеги. Я прошу прощения, у нас здесь проблемы были со связью. Я рад очень видеть всех своих преподавателей бывших. Спасибо вам. Я не буду вас мучить своим плохим английским. У меня 3 вопроса, я, наверное, пропустил. Как вы измеряли экспрессию инсулина?
Сразу задайте 3 вопроса, чтобы... Yes.
Да, тогда первый вопрос у меня. Как вы измеряли экспрессию инсулина? Второй вопрос. Какой метод вы использовали для подтверждения экспрессии инсулина? ПЦР или Western blot? Третий вопрос. Сколько гидовых РНК вы тестировали и как выбрали наиболее эффективные? Спасибо.
Should I answer in Russian or in English? Russian. Хорошо. Да. Ильяс Амирханович, спасибо большое за вопросы. Я, наверное, начну с третьего вопроса, который уже был озвучен Гульнур Сагандыковной, потому что как мы валидировали вообще, да, эти виды РНК. На самом деле там все просто. Есть assay, да, метод, который называется nucleus surveyor на английском языке, когда для того, чтобы выбрать наиболее эффективные гидовые РНК, нужно использовать нативную форму самой нуклеазы Cas9. То есть она активная, она работает так же, как и в бактериях. Получается, с помощью, соединяя гидовые РНК с активной формой нуклеазы, их внедряют обычно, если это мышиные клетки, то это или это человеческие клетки, островки клетки, да, островковые клетки. И тогда мы смотрим, насколько гидовая РНК может соединиться с нуклеазой и драйвить нуклеазу в нужную для нас таргетную последовательность. Если же после, в моем случае, когда клетки островков перестали вырабатывать инсулин, получается, в данном случае гидовая РНК работает отлично, и эту гидовую РНК можно использовать в дальнейшем для уже своей работы. То есть это вот специальный метод называется. Этим методом я использовал, и она, в принципе, прекрасно показала мне, какие гидовые РНК нужно дальше использовать. Но на самом деле программа, которую использовал, GuideMaker, она мне дала свыше 50 опций гидовых РНК, и это, получается, в моем случае как библиотека была, и из библиотеки я уже частично уже отбирал, какие гидовые РНК нужны. А в конечном итоге для уже конструкции я использовал 2 РНК, потому что, согласно литературе, сейчас до 4, если использовать до 4 гидовых РНК, то повышается эффективность самой конструкции в целом. Это на третий вопрос. Первый и второй вопрос, я думаю, они взаимосвязаны. Экспрессию инсулина я старался всеми возможными методами смотреть. Конечно, в данном случае не заменит никакой метод обычный ПЦР, real-time ПЦР, когда можно смотреть экспрессию инсулина. И второй метод для уже подтверждения экспрессии инсулина, Western blotting хорошо был озвучен, но с этим методом проблема большая, потому что инсулин, он белок всего лишь 151, да, аминокислот очень маленький, и я несколько повторений делал, и Western blotting, она в какой-то момент, я терял образцы, то есть из-за того, что она маленький образец, мне не удалось подтвердить. И потом я уже начал использовать стейнинг, окрашивание с помощью красителя Hirsch, да, и который как раз-таки показывает, какие клетки уже на уровне протеина вырабатывают инсулин. Но мне стало интересно, потом я стал гуглить, неужели никто Western blotting инсулин не детектировали, и оказывается, только в истории только одна лаборатория, эта лаборатория находится в Японии, как-то они умудрялись Western blotting улучшить, оптимизировать для того, чтобы все-таки, скажем так, поймать инсулин методом Western blotting. Но это было очень сложно, и она действительно из-за того, что размер маленький, большие сложности были. Но для подтверждения я использовал потом окрашивание, ну и также флуоресцентный микроскоп с помощью включения репортерного гена, в моем случае TSRED, конструкцию. Она показала именно, в каких клетках сначала транскрипция прошла, ну и какие клетки уже потом вырабатывали инсулин.
Спасибо.
Ну,
Ильяс
Рахмет,
Алифат,
ага.
удовлетворены?
Да, Рахмет,
Вам спасибо
да,
большое.
удовлетворен, Рахмет.
Спасибо. Коллеги, Алжанұлы, он вырос собственно генеральную систему учителя. Кроме вдруг родная подошли к чужому. Почему? Потому что технологический тест, сколько комплементарность промоутеров гена инсулина. Не так просто добиться комплементарности. Вот в этом, как вы видите, в чем суть комплементарности, которую можно привести. И почему перерождение. Вы знаете, иммунная система, она всегда больше, чем живот.
Да. Рахмет сұрағыңызға, Сұлтан Төлеуханович. Өте енді концептуалды сұрақ болып тұр ғой, бұл енді бүкіл қазір диабетпен ауырған адам алдында тұрған. Неге бір күнде өз жаңағы жақының былайша айтқанда енді не болып шыға келді деген сияқты. Оның себебі өте көп. Бұл жерде генетикалық фактор да бар, бұл жерде қоршаған ортаның факторы да бар. Әсіресе қоршаған ортаның ішінде мына инфекциялардың рөлі күшті болып тұр. Осыдан 2-3 жыл бұрын Қазақстанда балалар арасында корь қатты таралып кетті, егер естеріңізде болса. Сол кезде мен біраз осы балалары диабетпен ауыратын ата-аналарды білемін. Кейбір чаттарда отырған кезде, сол кезде корьдан кейін диабеттің бірінші типінің диагнозы кішкене көтерілгендей байқалды. Мен кейін мынау медициналық даналарды қарап көрдім, әлі обновительный шығып қалыпты. Сондықтан бірнеше себеп бар. Әсіресе енді вирустық инфекциялар жаңағы триггерующий фактор ретінде көп айтылады. Аяқ астынан бір күнде мысалға жаңағы ауто антиденелер түзіліп, жаңағы иммундық және лимфоциттер именно осы альфа клеткаларды емес, жаңағы аралшықтың ішінде өзі араласып тұрады екеуі. Именно бета клетканың жаңағы рецепторларын тауып, сөйтіп өлтіретін ол жұмбақ әлде.
Пакистанда, Россияда, Қазақстанда, Россияда. Екіншісі сол территорияның балансы. Енді ана ультрафиолет. Информация мәлімдейді. Ультрафиолет бұл қазір сол шарттық
Вива протоколды жаңағы инвиватор сатысы протоколды жасап, енді CRISPR-ды қолданбай, мысалы жай салды клеткадан протокол жасап, әп-әдемі клетка алуға болады. Оларда жаңағы глюкоза жауабы бәрі күшті болып тұруы мүмкін. Енді осыны ары қарай қазіргі көп тоқтап тұрған жұмыс ол инвивоға барған кезде енді бәрі былайша сноппына болып өзгеріп кетіп жатыр да, ана бәрі сондықтан сол жерде болып тұрады. Бірақ ана жаңағы компаниялар, ана Vertex деген олар қазір жаңағы қолдан искусственный қорғаныш қабат ойлап тауып, ішіне қан тамырларды жіберіп, максимально кәдімгідей нативный среданы мимик жасап, сөйтіп соны екінші этап клиникалық фазаға жеткізіп қойды. Олардың CRISPR-мен де алынғаны бар, екіншісінде CRISPR-сыз жай алынғандар бар. Негізі сайтомегаловирустың промоутері жақсы жүреді, ана екеуінің фьюзл болғаннан кейін екеуі де екеуі де ана VPS
Так я удивляюсь, почему инсулин поэтому краб лучше будет подавлять экспрессию, ладно, свою P64.
иә бір кішкене точнейший момент VP64 болған кезде ол ана как раз краб как инструмент болып тұр ғой, краб гайдеренкаға жабысқан, гайдеренкаға VP64 жабысқанда екеуі промоутерға барып отырған кезде VP64 транскрипция факторлар шақырып, иә краб мешает VP64 иә екеуінде де бірақ
да енді краб белок BP64 жұмысына кедергі жасайды, оны түсінбейсіз.
ана
мен очень таңдалдым, инсулин талап айтады.
конкуренция
Краб контроль интенсивный промоутер. Когда надо. Может, он даже инсулин очень разный.
негізі жақсы сұрақ. Мен айта кететін болсам, негізі ана жаңағы бағаналы жасушаларға барған жердің барлығында мен VP64-мен жүрдім ғой, арттырып алдым да, сонымен ары қарай жүрдім. Енді мен крабты қосқан себебім, мен тек крабты жаңағы обычный хэк клеткаларда, адамның клеткаларында уже арттырылған экспрессияның үстіне келіп крабты дикаснай мен басқамен қосқан кезде ол ананы түсіре ме түсірмей ме деген көрінеді. Өйткені ол үшін ғана түсіретін себебі екеуіне гайдеренка бірдей болғаннан екеуінде конкуренция пайда болады. Қайсысы көбірек или подавляет аха, қайсысы енді кім келіп қалай победит. Оларда краб жоқ, бағаналарға мен крабты кіргізген жоқпын.
Экспрессиясы третий вот. И тогда он такие пушки. Он начинает реагировать на это. Тоже пару пошел, дару помой. Там он независимо от того, интеграция, каким образом образовался
енді бұл жерде ана бағаналы жасушалардан енді біз инвитро жағдайында ұйқы безінің клеткаларын алдық деп отырмыз ғой, биомаркерлер бәрі сәйкес келіп тұр. Сондықтан ол жерде енді мынау ішінде жаңағы CRISPR-дың қалып кеткен несі, ол мына жаңағы клеткалардың біреуінде өзгеріске ұшырайды бәрібір. Енді сол нақты жауап жоқ бұл жерде, қалай жаңағы глюкозаға жауап пайда болды деген. Бірақ бұл жердің пока мына feasibility study болғаннан кейін не болады екен деген көріп жатырмыз ғой. Бірақ сіз қойған сұрақ ол бар, кішкене болып тұр аха. Енді
он же не зависит от чего-то промотора. Каким образом появится? Видишь, что с Вестерном всегда с этой вроде инсультиции. Нанограмма. Означает, что количество.
Количество, ахауыз ана Бетфорд.
Если вы Вестон будет нанограммы. Означает, вы использовали метод количества.
Количественный. Бетфорд метод деген бар, Бетфорд деген ғалымның атын ашқан. Ол Элизамен көріп, бірақ қазір мен protein quantification дейді ғой. Мысалға бұл жерде жаңағы клеткалардан 3 түрлі концентрация глюкозаға ұшыратқаннан кейін бүкіл ана бірінші клеткалардың барлығын протеинді бөліп алу керек та. Жаңағы неге глюкозаны қосқанға дейін, сіз қосқаннан кейінгі протеин екеуін бөліп салыстырып, сөйтіп протеин quantification мен жаңағы пиктарымен көреміз. Сол кезде сол Бірінші жоқ общий белокты бір көреміз, инсулинмен де бір көреміз. Бірақ ол антителомен анықталады. Элиза деп айтылады. Иә, антителомен.
I also have several general questions. Well, first of all, thank you for your very nice presentation. I wonder, in the world there are many groups studying this subject. In your perception, what's the novelty of your study? Can you list the things you think it's novel for this area of your study?
Well, the novelty lies in the point where I used CRISPR technology first to modulate stem cell, insulin expression stem cell, because there were lots of studies done to modulate endogenous gene expression in stem cells, for different genes, but it was specific for insulin. For my interest and for my wondering, there were very less studies done on insulin gene using, genome editing technologies. So for me, it was interesting to see how the CRISPR technology would allow me, in pair with synthetic transcription regulators, to modulate insulin. So for that, I can say it's a novelty, but not quite novel. But the novelty was that the differentiating CRISPR-edited stem cells toward pancreatic beta cells. I would say that was the main part of the novelty of my research. And being able, if I may say, able to see that at the end of the differentiation, I still have some detectable level of insulin expression in the differentiated cells, because many CRISPR constructs, zinc finger constructs, they are lost during the differentiation. Because differentiation, that we add many types and we expose cells to many growth factors, so it's very significant changes. Yeah.
The second question is, your work is sound technically perfect. It's just lots of different tools using. How about the possibilities to do this research in Almaty, in this scientific community? What's the possibilities to do this work? How well could your organization and maybe some relative organization kind of small? How do you assess the general advancement of this kind of study here?
Good question. Thank you for this Yerlanking speakage. Before going to Canada and before doing these manipulations by my own hand, I was thinking that those type of researchers probably they're not going to come to Kazakhstan. I was imagining that they're super high-tech studies, but in fact, it's not quite difficult for me, because I was able to I did most of the research in just 5 months, and so now my plan is that my research grant for Shatphone competition was already granted. Of course, the money is quite low for that, but for future grants, I would like to, with the help of Komalit Orumbayevich, to try to do at least some part of the research with cells, of course, because the qPCR can do in every scientific institution, but at least with cells to do some manipulation with fluorescence microscope to play around. I do have plans to.
And my last question about what do you think about, or your vision, how to convert your study for the practicality? With some question about practical applicability. So any environment where you can enter and introduce your work and to be successful in this field together with the practitioners.
That's a good question, and it's a very logical question. But as I said, in order to talk to practitioners, there is a long path to go through yet. Because even if, for example, let's imagine that the cells are responding to glucose very well, But there is another question. Of course, it's just a feasibility study, but in vivo, how I'm going to control my constructs? The mouse in vitro, I can transfect, transduce, and see the RNA, but in vivo, I need to somehow control when the crab protein when to act, and also tell the VP64 when to act. So for that, there is one possible solution, which is called optogenetic, when by applying a special light of a special mostly it's a blue light, which is 490 nanometers. I think the in that case, there are some proteins when exposing to blue light, the proteins they dimerize, and with that there is some manipulation possible to do in order to control all these constructs. So even let's say if I'm able to do that, then still in vivo and in later stages of the research, it's still going to be challenged. So there is long path. But also, of course, considering about the practical realities.
Okay, dear colleagues, any questions? Maybe you have still some questions left. If no, then thank you very much, Mahchan. So you may sit down and we will move to the next.
Can I ask a question, please?
Yeah, please.
Yes.
Mahchan,
Thank you very much for your presentation. I got a good question about tricky word in the title of your thesis, PhD thesis. I got a question about tunable. Engineering a tunable insulin production in beta cells. What do you mean by saying tunable?
Thank you very much, Dauren Adilovich. That's a very good question, and I was actually expecting this question from anyone. So the word tunable in this research is more related to using the transcription activator, and then I used another construct to lower that already activated level of insulin expression. So it's kind of tunable, but it's not 100% related to that, because in tunable, it's kind of like switchable. So in order for it to be switchable, was I already said the optogenetics using the light to switch, I mean, in a very fast mode, that would be more applicable. So, because I'm already planning to continue this research with optogenetics, so I'm still using the word tunable, but in this case, it's still related to VP64 and crab proteins, because one is activates and then it's another inhibits, so it's kind of tunable.
okay.
But it's not actually the word that you should use for this kind of, because it means you have to tunable, you have to use in the same cell type. You have to be or express or depress the protein production in the same cell. So it means tunable. So actually not really like a, let's say, totally correct to use this word in a type of cells that you produce. So you use two different kind of cells. But and it means so tunable, it must be like a switching on and off when you need it.
Okay.
Then I got another question about, so this work actually, so I sorry, but maybe I missed, but did you eventually get your differentiated cells or not?
Yes, I did receive my differentiated cells.
So you use like a H1 cells or HEC cells. You use the like a gene modification technology, and you get your differentiated cells that produce your insulin. So you reach the goal that you wanted. But
yes,
have you tried or have you planned to differentiate the HEC or HEC1 cell or not H1, the HEC cells that are like a natural cells, as I assume, right or not?
Yeah, the HEC cells, they are ordinary human cells. They are not stem cells. But the question is interesting. Why not to differentiate them towards stem cells and then try to differentiate them?
And then use your gene editing technology. Try to, because they meant to produce the insulin. The cells. So it will give you opportunity to test the levels of expression of your, cells, let's say, pluripotent cells. Let's say you did a transfection, you did the differentiation, or you just did a differentiation and then you did a gene editing.
Yeah, if I understood correctly, your questions, there are two questions. One is related to HEC cells. I mean, they are ordinary human cells, so they are not stem cells. The HEC cells are used just to validate the effectiveness of my construct, to understand if I'm doing right or not. When I see that,
that's very interesting question. Of course, that's Alimjan. You should explain that's your private opinion. That you have opinion. Please explain.
Yeah, thank you. Yeah, so that's I haven't thought about using HEC cells, ordinary human cells, to differentiate them towards stem cells, then try to use them for something else, which is very interesting and very quite a rare question, but still question. I mean, thank you for that. But in my case,
is it promising?
Yeah, but in my case, I used H1 stem cells. First, I changed the transcription of insulin first, and then I differentiated them towards beta cells.
Because, but there's actually, so the gene of insulin, natural gene, is still present in H1 cells, or it's absent?
No, it's there, but it's not switched on yet.
Yeah, but so I mean, there was okay. Then I will rephrase my question about the using H1 cells, that you do the modification before you differentiate them, and then after they differentiate.
I did the modification before differentiation.
So how it will how your construction will affect
My question is because there's a clinical that's you have any application for clinical work? No?
Now we have a question from Dauren Adilovich. He is answering the questions from other reviewers and from Dauren Adilovich.
I'm understanding that just for future.
Thank you.
Thank you.
Yeah. Let's see how Bakhjan replies to the question of Dauren Adilovich.
So Dauren Adilovich, so in the case of H1 stem cells, I first modified the genome in terms of modifying transcription, then I differentiated them.
Because your construction consists of activator, if I'm correct.
Yes, activation construct.
Did you try to use how does this activator will influence the natural gene in the H1 cells that were differentiated to the better insulin producing cells?
Yes, exactly. I wanted to first activate the level of insulin transcription in stem cells, and then during the differentiation, I wanted to see if the effectiveness of the construct is going to be lost or not, and if lost, for how much. That was the research question in my case.
Okay.
Thank you, Dauren Adilovich.
Thank you very much. I'm satisfied with the answer.
Janat Ayhanovich, do you have questions? Окей. Ну, anyway, в любом случае, мы
No any question. Нормально. No any question.
Bakhchan, please sit down. We will move to the next stage. Next stage, we should your opinion, supervisor, пожалуйста, Врач, ваше мнение как руководителя о вашем студенте, если можно кратко, вы видели, сколько много вопросов было. Да, надо сюда, чтобы не слышно. Алжирский вариант давно здесь?
Thank you very much, Martin Speaks, for the opportunity to express my opinion to my doctor, Dr. Бақытжан Алжанұлы. Бақытжан Алжанұлы жаңа айтып кетті, 2013 жылы осы университетті бітіріп, алтын белгімен ол да оңай емес. Одан кейін Америкада жаңағы болашақ бойынша оқыды, 2017 жылы оны бітірді. Одан кейін ары қарай жұмысын жалғастырды, Ванкувер университетінде істеді. Осы уақыттардың бойында Бақытжан Алжанұлы осы тақырыпты, бұл тақырып өте өзекті, ары қарай жалғастырып отырды. Енді тақырыптың өзектілігіне екі-ақ сөз ауыз айтайын. Жаңа әдемі сұрақтар болып жатыр. Жер жүзінде қант диабетімен ауыратындардың саны 2-10% арасында, әр елге байланысты. Біздің Қазақстанда где-то 3-3,5% адамдар қант диабетімен ауырады. Бұл жер жүзіндегі мәлімет бойынша орта көрсеткіште тұрмыз. Кейбір елдерде, мысалы Монғолияда қант диабетімен ауыратындар болмаған, бірақ соңғы 10 жылдықта мынау батыстың құндылығымен фастфуд жеп, оларда қант диабетімен ауыратындар пайда бола бастады. Бұл өзекті мәселе, әсіресе бұл өзекті мәселе жаңа айттым, 3-10% деген өте үлкен халықтың, мемлекеттің экономикасына қаржыға түсіретін үлкен қасірет. Қасіреттің ең жоғарысы сол, что бұл кезде қант диабетінің бірінші түрі пайда болады. Адамдардың ауруы қасірет, жаңадан туған нәрестелер 3 жастағы балалар ауырған кезде ол 2 есе қасірет. Сондықтан бұл жұмыстың өзектілігіне ешқандай күмән келтіруге болмайды деп ойлаймын. Разрешите сказать пару слов. Личный вклад докторанта Бақытжан Алжанұлы. Работа является относительно завершенной. Бақытжан Алжанұлы созданный дизайн направляющий ДНК, нацеливанный CRISPR-комплексы, промотор гена инсулина на основе лентовируса создан, вектор для доставки выбранных РНК, таргетные клетки и другие технологии. И все эти методы в основном разработаны лично Бақытжан Алжанұлы. Это его дизайн, это его конструкция, это его мысли, это его работа, вот эту общую работу вести. И также хочу сказать, что учитывая личностные качества, Бақытжан Алжанұлы не только его ценности направлены на фундаментальную и прикладную науку, я как руководитель могу говорить, что он как человек обладает очень скромной, стрессоустойчивой, это главная, я думаю, позиция, и самое главное, трудолюбивый и целенаправленный, мотивированный человек. Я считаю, что если вы поддержите, его работа на определенном стадии завершена, он заслуживает присуждения степени, просим вас, PhD доктора. А также сейчас просто 2 слова скажу как руководителя, как эта тема меня тоже волнует и волнует наших врачей, вот, Жана Айкановича и других наших диабетологов. Вообще-то мы достигли определенном уровне те технологии, которые на Западе были разработаны. Мы сейчас его транслировали, трансфер технологий, освоили, немного модифицировали. Но вопрос стоит сейчас в другом. Эти технологии освоены. Теперь вопрос стоит, как регулировать выделение и отключение свечов или нейтронов, то есть включение этих клеток. В организме человека так создано, что когда концентрация глюкозы в крови повышается больше, чем 5,6, он автоматически включается, когда падает там 2-3, автоматически выключается. Это в организме так прекрасно, ретроингибирование работает по глюкозе. А как теперь, если мы создадим эти с использованием стволовых клеток, как будем отключать, включать? Есть работы по оптогенетике. Я думаю, в моем мнении, это не совсем актуально. Надо по-другому подойти. Даже если мы будем копировать западные технологии, мы будем отставать. Нам надо с Бахчаном и другими единомышленниками, вы помогите. Надо другую технологию подумать, чтобы как включить, подключать, когда поднимается, снижается концентрация глюкозы в крови. Спасибо за внимание. Спасибо, коллеги.
Спасибо большое, коллега. Second advisor, Dr. Pajinski.
The review of the foreign scientific supervisor for the research work Алжанұлы Бахчан is positive.
Уважаемые коллеги, у меня просто одно замечание. Самое главное, любая технология, которая поможет нам победить диабет первого типа, будет как никогда уместно.
Спасибо.
So, being a promising early career scientist, Bachchan practices a professional attitude in conducting scientific work. He treats...
No. I can speak directly. That any technology for treatment diabetes type 1. Just super.
For the advisors, Bachchan Alžanұlы's advisors, foreign advisors. She is just reading her review of the supervisor. Thank you.
He treats tasks with full dedication and constantly acts as an initiator on optimizing the workflow and an active participant in scientific discussion. It was a pleasure working with him alone while he was visiting us, and since then we have been in close contact. I hope for more collaboration with him in the future. Therefore, having said that above mentioned, I believe that Bachchan himself and his research work deserve the degree of philosophy doctor in the specialty biology.
So now we are moving to the official reviewers of this work, and now it's time for Janaya Kanovich to briefly introduce your opinion on this work. And if you have any comments or questions, please ask. Now is the time for your assessment of this work. Please.
Thank you very much. Thank you.
Жанай Камич, можете включить видео? Вас не слышно, не видно.
Unfortunately, I cannot use the video. Sorry.
Please continue.
Just actually speaking.
Okay.
I'm so sorry. Actually speaking, I think that this research is very promising, and we should try to support this research. This is my private opinion.
Do you have any questions or comments to this work?
I have no any question. I think that this research is a very promising. Is the first step to the next step.
So your official answer would be you are supporting this work, right?
I think that we should support this research.
Okay. Thank you very much for your opinion. And now we providing the space for second official reviewer. Ережепов Дorian Адилович, пожалуйста, вам слово.
Thank you very much.
Спасибо большое. I will not be so very, how to say, long saying. So I found out that this work is done in a part of the work done during the grant funding. So the self-reliance is high. The relevance of this is justified, and the scientific novelty of the principle I put partially new, because it's not like a work that's done in a worldwide. So there are many laboratories that have the same like similar works. So and I want, do you hear me?
Yes, yes, please continue.
Okay. And I have one thing to say, that it's a very good work. However, it represents only one peer-reviewed journal, the experimental part of this research, which is not included in the Web of Science database, but presented in Scopus, and by the Scopus, like a level of the journal, it is passes the how to say, passes the minimal like requirements for the dissertation. And another thing, that the source of the HEC 293 cells in your thesis and in the how to say, in the article are different. So you like put different sources. And additionally, there's no joint publication with your foreign supervisor. So, which does not demonstrate the supervisory contribution of the latter. So there's no joint publication. So I cannot estimate how much you worked out there, what did you do together. So, but in the end, so I got decision to award the degree of PhD degree to you, because this is a really good work. This is a really good, like how to say, like one of the breakthroughs. If it will be implemented in our country, it will do a difference.
Okay. So your final answer would be positive. You're supporting this work?
Yes, I support the work.
Very much. Now it's time for Bachchan to comment both reviews, both with other cases. We have a few minutes to comment and put something about official review management.
Thank you very much. I thank both Daurin Adilович and Jana Ikanovich for their time and for their efforts in evaluating my work, assessing all the details.
I'm here.
Thank you very much, Jana Ikanovich. Thank you. Daurin Adilовich, questions. Your questions are very good and very detailed. So if I start from the sources of the HEC cells that I showed in the dissertation and in the article that was published in 2001, they really do differ, because for the article, the cells I used, that part of the work, that was a repetition of the one part that was already done abroad. So for that work, I used the HEC cells from the Aspegar University Collection Center. But for the dissertation, the first part, the validation part, since it was done in Canada, I used the cells that was brought from the facility of the University of British Columbia. My answer for that. Second answer is a very interesting question. Really, I don't have yet a joint publication with my foreign scientific advisor. You know, it's simply because in this area of coming up with a therapy for diabetes type 1, there is a big race going on worldwide. I mean, there are lots of research groups and labs. They are really competing on this area. And the main question is, who will be the first to come up with a protocol that will be, you know, functional and that will be repeatable, etc. So in terms of the differentiation part of the protocol, my scientific supervisor's position is very strong, and he wants to be his next publication in this area be on protocol. So any publication before the protocol, he doesn't want to include his name. So he kindly
You should be.
Someday I hope we will have a joint publication, but for the experiment part of the results that I published, my scientific supervisor, he kindly let me publish that part of the work, but without his name, because as I said, he wants to have his name be only connected to the protocol, which is coming very soon. So that's another reason, because that's a right of the researcher, scientist, where to publish his name. So that's my honest answer for your question, but that's a very good question you mentioned.
Thank you very much, Lucy. Now we move to the next very important step of the defense. It's a discussion, and we'd like to hear first our invited members of the scientific panel. Roland Bogdanovich, can you please tell about your opinion on this work in general?
Can you hear me?
Yes.
Dear colleagues, dear committee, dear Bakhcan, thank you very much for this interesting work. As we discussed, this is a promising area, and you are at the very probably the first steps of developing cellular therapies. And as you mentioned in your thesis, looking for donors are not always easy due to multiple factors. And in future, hopefully, these cellular therapies will really help, to apply in clinical practice. As was mentioned by Dr. Davron Yerojepov, there were a few things that probably need to be addressed. Especially, I also picked up the tunable word in your title, because just thinking about the baseline insulin expression, and tunable, by tunable we mean adapted expression depending on the environment, right? Where insulin is needed, it can switched on, and when it's not, it can be turned off. And I wonder if these things have been addressed in the thesis. If not, maybe it's better to remove tunable. I mean, this would be my advice. But in general, I really liked your work, and I can see that you have done a very hard work, and I fully support your entitlement for a PhD. Thank you very much.
Thank you very much, Raul Bogdanovich. In general, we thank you very much for attending this PhD defense and for your contributions. Thank you. Colleagues, please, if you have any opinion on this work, you may express your opinion right now. Just raise your hand or turn on your microphone.
Слышно, да? Слышно? Если слышно, работа очень, я думаю,
Да,
живет.
слышно.
Ну, с использованием самых современных методов генной инженерии. И получены очень хорошие результаты. Вот сейчас только научный руководитель команды Владимир Павлович сказал, что с вашей, то есть помощью хотим дальше продолжать данную работу. Я поэтому хотел дать свое мнение, дальше как продолжать. Я на самом деле интересовался данной работой. Оказалось, что именно с помощью такого метода созданные стволовые клетки, дифференцированные, да, в бета-таких, уже проходят клинические испытания третьей фазы. Вот как они сделали? Они, во-первых, использовали не эмбриональные стволовые клетки, да, а фибробласты кожи. из них сделали индуцированные стволовые клетки, так, с помощью 4 или 3 там, транскрипционных факторов. Таким образом получились стволовые клетки. Затем они не индуцировали экспрессию инсулина, они начали индуцировать дифференциацию стволовых клеток в бета-клетки. Каким образом? С помощью 3 регуляторных генов МАФа, MTX1, да, например, да, и эти еще транскрипционные факторы, как только получили дифференцированный клетки, инсулин сам по себе начал синтезироваться. На самом деле регуляторные гены как раз и нужны для того, чтобы индуцировать инсулин. Это во-первых. Во-вторых, таким путем сделаны бета-клетки.
Не соглашусь.
Больному это аутоиммунное заболевание, да, первый тип диабета. Он в любом случае иммунитет уничтожит просто. Поэтому они что сделали? Чтобы иммунитет не уничтожил данные, кроме такого характера модификации, который я отметил, да, они есть такой главный компонент личной совместимости. Да, у человека это называется HLA или Human Leukocyte Antigen. Да? Их 20 до 200, они гены классные. Это как раз указывает, эта клетка стоит атаковаться, не стоит. Они как раз указывают на внутриклеточное состояние клетки. Они там миллионы белков, если да, они пептиды берут, и на поверхности клетки они стоят, торчат. И по ней узнают лимфоциты, начинают атаковать, и антитела связываются. Они нокаут сделали всех HLA генов. После этого иммунитет уже не атакует. В-третьих, еще сделали, что повесили на поверхность этих сконструированных клеток так называемый ПДЛ-белок. ПДЛ-белок это называется программ cell-based ligand. Меченные ПДЛ-белком клетки, на самом деле, да, они атакуются Т-лимфоцитами. Потому что на поверхности лимфоцитов сидит PD-белок. PD-белок связывается с ПДЛ, да, тогда он думает, что я допустил очень серьезную ошибку, начал атаковать очень серьезный, он запускает апоптоз. Наоборот, иммунные клетки запускают апоптоз, умирают. Вот таким образом они сконструировали бета-клетки, которые в настоящее время проходят клинические испытания 3-й фазы. Но это мое мнение относительно того, в каком направлении данную работу надо развивать.
Не согласен.
На самом деле работа очень хорошая, на самом деле, да. С помощью Cas9 и VP64. Сейчас в данное время VP64 это искусственный белок, да, и от VP16 белка, да, вируса герпеса. А сейчас значительно улучшились транскрипционные факторы, так называемый SAM белок. Да, например, он индуцирует транскрипцию любого гена значительно лучше, чем VP64 белок. Поэтому, так на самом деле очень хорошая работа, да, на высоком уровне. Последующим, мне кажется, у меня такого характера советы, в каком направлении развивать данную работу. Я считаю, что работа достойна. Доктор.
Могу я просто мое мнение.
Ахмед,
Жанарий Александрович, пожалуйста.
Мое мнение, любая технология, которая поможет в диабете 1-го типа, она приветствуется.
Көп рахмет сізге, The colleagues here, offline members, all supporting this work. How about online? Do you have any additional opinion? Gulnur Sakundekova? Some other guys? Ильяс Амирханович. Ильяс Амирханович. Или достаточно? Ну, тоже тогда скажу.
Да,
Да.
Илья Самирханович, мнение будет у вас
Я-
общее?
Не, ну работа вообще интересная. Я вот когда читал, очень понравилось. Диссертационная работа является завершенным научным исследованием, обладающим высокой степенью актуальности, новизны,
Диссертационная
практической
работа
значимости.
является
По своей
очень.
структуре, содержанию. Алло,
Да-да-да,
что-то у меня
слушаем.
связь.
Мы слушаем вас,
Нет,
слышим.
работа хорошая.
Диссертационная
Работа
работа.
хорошая. По
Спасибо.
содержанию она соответствует требованиям, предъявляемым к диссертации на соискание степени доктора философии
Да.
и заслуживает присвоения соответствующей ученой степени.
Спасибо большое. Спасибо, очень ценное мнение. А, коллеги, мне как председателю нельзя высказывать свое мнение, потому что он будет ангажированным. Поэтому мы переходим к последней стадии нашего заседания. Оно самое ответственное. Мы должны ответить электронным образом на вопросы, достоин ли Бахчан, фамилия, Алжанұлы, степень PhD, или еще какие-то там есть опции есть. Поэтому сейчас Мирапуль Кудретовна разошлет всем электронные формы, на которые вы ответите соответствующим образом. Там будут 4 опции. Пожалуйста, выберите одну из них и, значит, кликните на то, чтобы принять свое решение, и оно электронно, то есть автоматически уйдет на решение нашего диссертационного совета. Я не буду говорить все это на английском, поскольку я вижу все здесь русскоговорящие, казахоговорящие коллеги. Спасибо всем. Так, еще раз скажите все. Да, все, кроме Амагира Хуабаевича, должны проголосовать. Итого у нас должны быть 8 членов сегодняшнего заседания. 3 постоянных членов, 3 приглашенных и 2 рецензента. 2 рецензента голосуют. Жанай Ахадович, Давай Надилович.
Хорошо.
Сейчас, секундочку.
Я на связи. На связи.
Вам надо проголосовать электронно.
Да, конечно. Мгм.
Сейчас я вам отправлю. Секундочку, подождите. Там, сейчас. Интернет немножко завис, сейчас я вам отправлю. Уважаемые коллеги, проверьте, пожалуйста, ваши почты. Прошу вас попросить.
Вы все должны получить сообщение или на электронную почту, или на WhatsApp, и должны проголосовать. Спасибо. Голосование зайдет в 5,
Не получил пока. Не получил.
Технический перерыв на 5 минут. Сейчас мы проголосуемся и потом уже снова соберемся для того, чтобы подсчитать ответы.
Я не получил пока. Не получил.
Вам на WhatsApp отправить сейчас.
Хорошо.
Dağız verdim.
Так, нет.
Шикарнейшая работа. Вот, на самом деле. Шикарнейшая работа. И перспективы шикарные дальше.
Ана Эканович, алдыңыз ба?
Извините, я это вслух? Перешлите.
Мы вам на WhatsApp выслали, посмотрите, пожалуйста.
Да, сейчас. Да, сейчас, секунду. Да, получил. Блин, на почту. Так. На WhatsApp. Скиньте на WhatsApp. На WhatsApp, если можно. Если можно. На WhatsApp.
А вам послали на WhatsApp уже?
Не вижу. Не вижу.
Сказал.
Да, получил.
Все хорошо.
Объясните, что там.
Да. Пройдите по ссылке, там будет 4 ответа, одну из них выбирайте и все. И отправить. Внизу.
На ссылку нажимайте, там 4 вопроса появятся.
Я отправил. Коллеги.
Все, спасибо.
Спасибо.
Спокойно. Отправил.
Теперь мы приступаем к следующей стадии нашего заседания. Миракуль Кудретовна, пожалуйста, продекларируйте результаты голосования.
Хорошо. Сегодня в голосовании участвовали 8 членов комиссии, и все единогласно проголосовали за присуждение степени доктора философии. Вот. Да.
Congratulations.
Членов Конституционного совета поддержали вашу работу. Мы поздравляем вас. И для того, чтобы подключить, мы должны проголосовать таким способом решения этого тайного голосования. Кто за то, чтобы подтвердить результаты тайного голосования, прошу поднять руки. Кто за? Единогласно. Если кто-нибудь против, нет, единогласно. Теперь же официально
Спасибо.
мы поздравляем вас и ваших руководителей, коллег с этим замечательным событием. И мы даем вам слово для того, чтобы вы вашей работе.
Да, уважаемые члены диссертационного совета, уважаемые приглашенные
Я
эксперты,
поздравляю вас.
студенты. Спасибо, Жанна Иканович, вам тоже, в том числе, за ваше время, за то, что уделили время, посоветовали, рекомендацию давали. И спасибо за вопросы, за сегодняшнее заседание. Спасибо.
И теперь последний шаг нашего заседания. Мы должны принять заключение по данной работе. Заключение было разослано всем членам сегодняшнего заседания диссертационного, включая рецензентам. Поэтому предлагается взять за основу это заключение. Если будут какие-то комментарии, коррекции, пожалуйста, просьба выслать ваши замечания на адрес Мейрамты Кудретовны на электронную почту, и мы учтем эти ваши предложения для того, чтобы принять заключительный вариант заключения. На данный момент предлагается принять вот эту текущую версию заключения для того, чтобы справиться с этим последним шагом. Прошу проголосовать, кто за то, чтобы принять эту версию заключения базовую. Если против, единогласно. Уважаемые коллеги, таким образом на сегодняшний день мы выполнили все шаги нашего заседания, все этапы. Я благодарен всем коллегам, которые приняли участие в сегодняшнем заседании. Здесь, в Казахстане, за рубежом, в Алмате, кто нашел время и участвует в онлайн-совещании. Большое всем спасибо. Надеюсь, это не последняя наша совместная работа. Надеемся, что вы также будете активно поддерживать диссертационный совет в плане оценки диссертационных работ наших PhD-студентов. Еще раз большое спасибо и всего наилучшего.
Спасибо большое.
Рад был всех видеть. До свидания.
До свидания. Сауымызды аяқтадық. Рахмет көп.